Thank you for a very generous introduction. It's really been great visiting this department, seeing old friends and making new friends here. And so today I'm going to talk about some of the recent works we did in our group. And so please feel free to interrupt if you have any questions. We know that genome-wide association studies has been there for a long while. And GWAS have identified many SNPs that are associated with complex traits and common diseases. However, even though you identify so many associations, it's not easy to understand why this association, what's the molecular mechanism underlying association. And so one hypothesis is that many of those SNP associations are sitting in non-coding regions, so they won't really change the sequence of DNA, protein sequence of the gene. However, they may affect the traits through gene expression regulation. So therefore, because of the hypothesis in parallel, in addition to the genome-wide association studies, there are many studies have been carried out to measure, in addition to the GWAS, there are many studies to carry out to measure gene expression, along with the SNP data to carry out so-called EQTL mapping studies. And the EQTL mapping studies have revealed that SNPs are indeed associated with gene expression. So therefore, you have two different types of studies. You have GWAS, you have EQTL mapping. And so naturally, people want to integrate between these two in order to really understand, looking into the molecular mechanism underlying disease etiology. And so there are two commonly used approaches to do such integration analysis. The first one is called PracticeScan from Harkey's group. They are marketing this as a SNP aggregation approach. And it consists of two different steps. In the first step, you look at the EQTL data, and you can, let me see, I probably forgot to open this. Okay, hopefully it's louder now. In the EQTL data, they try to construct the genetic predictor of gene expression using ElasticNet. So that's step one. The idea is that you have SNPs, you have gene expression, while using SNP to predict gene expression. And then in step two, since now you know the SNP weights for predicting gene expression, you are going to plug in the weights in the GWAS study, multiply by their weights. Then you can construct this so-called the GREX, or gene regulatory expression, and GREX here, genetically regulated expression. And then once you construct this GREX, you can do association tests between the GREX and the trait. And so this way, you can test the association between the genetic predictor of gene expression and trait. So it consists of two different steps. And it's using the ElasticNet to construct gene expression prediction. So that's the first method. And the second method is TWAS, or transcriptome-wide association study. And it's marketed as expression trait associations. And it also consists of two different steps. In the first step, it's also looking at the EQTL mapping data. It's using the cis EQTLs to construct a predictor of gene expression. And this time, it's using BSLMM to construct this prediction instead of ElasticNet. Then in the step two, once you have the SNP weights predicting gene expression, now you can plug in the weights in your GWAS. And you can construct the GREX again. You can test the association between the genetic predictor of expression and the trait. So you can see that both methods consist of these two different steps. Both methods looks almost identical, with the only exception that one uses ElasticNet to construct gene expression prediction, the other uses BSLMM. BSL is based in sparse linear mixed model. Yeah, it's sort of well known in the genetics literature. Yes.
So the two methods differ in only the method that's used to predict the gene
expression. Exactly. Yeah. So that's the only difference, and there's two separate Nature Genetics papers. And so once you see this two-step approach, you can clearly see that in this two-step approach, because you are doing step one, step two. In step one, you are constructing prediction of gene expression. But in that prediction step, the SNP weights to predict gene expression, it actually comes with a lot of uncertainty because you have a small sample size data in gene expression. So the SNP weights has a lot of uncertainty in the step one. However, the step two never really accounts for such uncertainty. So naturally, you would wonder whether if you fail to account for that uncertainty, maybe you'll lose power in the analysis. And second is that for those people who are more familiar with the statistical or causal inference literature, maybe you will quickly recognize that this two-step approach is basically a two-step Mendelian randomization, where the Mendelian randomization is an instrumental variable method for causal inference. So therefore, and from a Mendelian randomization perspective, you can clearly see that
both methods. First, they fail to account for the uncertainty, the gene expression prediction uncertainty, but also second, perhaps even more importantly, they fail to account for the so-called horizontal pleiotropy effects, which turns out to be quite widespread in TWAS applications. I'll show you later. So to see this more clearly, I think it helps to take sort of step back to see how people generally do Mendelian randomization to do this instrumental variable analysis for causal inference. So in this setting, right, in the causal inference setting, we have our gene expression was simply denoted as X. And we have our complex trait. It could be diabetes, it could be height or BMI. And our goal is to infer the potential causal effect of gene expression on the trait. So that's the alpha effect. So the alpha effect is our key parameter of interest. So to do this, if you collect gene expression data and trait data in the same data set, you can easily do a correlation analysis to do that. But one issue with correlation analysis is that there's always some confounding factors that's affecting both gene expression and the trait. You can imagine that if you measure gene expression trait in the same individuals, there might be batch effects. There might be some other confounding factors affecting both. So therefore, if you do a simple correlation analysis, it won't give you causality. So therefore, people can try to introduce a different set of variable. They're called instrumental variable. And in this case, those are the SNPs. So they try to treat the SNPs as instrumental variable. So specifically, if the SNPs are associated with gene expression, then you can treat them as instrumental variable, perform instrumental variable analysis. And through this way, you can basically bypass the confounding factors, allows you to really infer the causal effects of gene expression on the trait. So this is really ideal. However, those instrumental variable analysis requires you make three key assumptions. So the first assumption is that the SNPs are associated with gene expression. This is usually satisfied in real data practice because you identify cis SNPs that are usually associated with gene expression. The second assumption is that the SNPs does not affect traits directly, but any SNP effects on the traits has to mediate it or go through the gene expression path. So that you can argue that perhaps this largely sort of satisfies this is sometimes it's not easy to argue whether it's satisfied or not. So this, you can see that it's connected as this path here. So the SNPs sometimes maybe will have effects on the trait without going through this particular gene expression, because you can imagine the SNP may affect other genes in the local region. SNP may affect methylation, maybe affect attack-seq or chromatin modifications in order to affect the traits. So therefore, it's possible that the SNPs actually exhibit effects on the trait, not through the particular gene expression. And this is a huge problem. And this effect people usually refer to as the horizontal pleiotropy effects, which are denoted as gamma over here. And in fact, in standard Mendelian randomization, where people look at the causal relationship between two different traits, they found that there's a widespread horizontal pleiotropy in causal relationship inference in Mendelian randomization. So this turns out to be a huge effect. So therefore, motivated by these limitations, what we're trying to do is to solve two issues. One is we want to control for such horizontal pleiotropy effects. Second is that we want to account for the gene expression prediction uncertainty. So to do that, we developed a method called PMR, or probabilistic Mendelian randomization. So the idea is this. So we're going to have two studies. One is gene expression study. You have SNP data, which are denoted as Z. You have gene expression denoted as X. We're going to look at one gene at a time, just as TWAS and parasitic scan settings. So we're going to build a model using your SNP data to predict this particular gene expression. So this is your first model over here. And then after you are able to predict this gene expression, right? So this is your gene expression data. And let's assume that this gene expression in the GWAS data, then we look at the GWAS data. So that's our second data. And typically in GWAS data, those individuals do not have gene expression measurement. So therefore, we will denote those gene expression measurement for each individual there as missing data, and we will simply denote it as X tilde. And we'll assume that the GWAS data basically consists of very similar in individual composition as your gene expression data. So therefore, this relationship between SNP to gene expression still holds in both in the GWAS data. So therefore, you can see that these two equations are identical to each other. So you have the same SNP effect size on the gene expression in the EQTL study, and you also have the same SNP effects beta on the gene expression in your GWAS study, except that in the GWAS, certainly X tilde is not measured. And then finally, we want to connect those unmeasured gene expression to the phenotypes. So if you are able to measure the X tilde, then you have X tilde over here. And the X tilde effects on the Y, that's basically a causal effect here. We denote it as your alpha here. And so this denotes that gene expression effects on the Y. And in doing so, you also want to control for that horizontal pleiotropy effects. So this term over here, Z times gamma here, the Z denotes the genotype. Those denotes the effect size that are not going through the gene expression, but directly onto Y. So therefore, by introducing the gamma term, now we are able to account for such horizontal pleiotropy effects. So that addresses our first issue, right? We can account for the horizontal pleiotropy effects. While on the second hand, in order to account for the uncertainty in gene expression prediction, basically the uncertainty associated with beta, then we also want to perform inference in a joint likelihood framework. So therefore, all the uncertainty associated with beta is accounted for. So therefore, overall, you can see that we have three different equations, and it's the beta that connects us through the gene expression study towards the GWAS. We assume that the SNP effects on gene expression are basically the same across those different studies. And certainly because the number of SNPs typically exceeds the number of samples over here. So we will need to make additional modeling assumption. And that's especially true in the gene expression studies, because in gene expression studies, we can easily have 1,000 SNPs, but our sample size is only 200, 300. So therefore, we need to make additional model assumption on beta. And for beta, we follow standard polygenic model and assume that beta a priori follows the normal distribution. So each SNP effect size follows the normal mean zero with a variance sigma square. And similarly for gamma, which is horizontal pleiotropy effects from SNP to the trait without going through the gene expression. We tried two different assumptions. One is also polygenic assumptions, the variance component version where gamma G follows the normal distribution. And the other one is we follow the aggregate regression. We assume that all the gammas equal to each other, and this also looks identical to each other, equal the same value. And this too also relates to the rare variance test. Specifically, the aggregate regression assumption relates to the burden test, while the variance component version relates to the scale test. And we'll primarily focus on the top, although both are implemented in the software. So that's our key model assumption. So we are able to account for the horizontal pleiotropy effects. And so in terms of inference, we use a joint likelihood inference instead of a traditional this two-stage or two-step approach. And because this way, if we jointly infer everything, then we can account for the expression prediction uncertainty. So the uncertainty associated with beta turns out it can help improve the power. And we develop an efficient algorithm based on parameter expansion EM algorithm using either individual level data or summary statistics. And we can test the causal effects on alpha through a likelihood ratio test. This is our causal effect. And we can also test the horizontal pleiotropy effects of gamma also through another likelihood ratio. So that's basically our model, the PMR model.
I'm wondering if you use summary statistics, how do you perform those likelihood ratio tests?
Yeah, so that's a great question. So it turns out that if you look at the three equations here, the joint likelihood is essentially basically it's a product of those different likelihoods. And it turns out that once you write down the likelihoods, you will see that it can be substituted with the summary statistic. Basically, one is X transpose X, the other is X transpose Y. So it's all considered just X transpose X, X transpose Y. And X transpose X is your marginal Z statistics. X transpose X is the SNP correlation matrix. So that's how we solve this. Yeah. Yes.
I'm not a genetic epidemiologist, so this question might be a little naive, but if there's this problem of horizontal pleiotropy, how do you identify that in the data? So what in the data set is giving you information to even estimate it?
Yeah, so that's a great question. So the data information is basically contains it's essentially all those data. So in the phenotype Y, in the data, you are able to decompose it into two components guided by your gene expression data. One component is your gene expression component. The other component is the non-gene expression component, but related to the SNP effects. So that way, once you decompose into two, you can see that the second part is your horizontal pleiotropy effects. The SNPs goes on Y, but not correlated with the gene expression at all. So I think that's intuitively that sort of explains where the information comes from.
And all data sets would give you that information.
Yeah, all the data.
Once you know how to partition this, the
Yeah,
information.
exactly.
So the information has always been there. We've never been looking at it.
Exactly. Yeah. Now we are able to do this partition effectively in order to extract that additional information. Okay. So we decide to look at the effectiveness of these methods. I think I'm probably going to skip all the simulations. Since in the simulation, they all work perfectly well, but I want to show you some real data results to see how it works. So specifically here, what we're trying to do is to use GWAS, which is the genes, the EQTL mapping study, and use the WTCC Wellcome Trust Case Control Consortium. It's a relatively small GWAS data set with only 5,000–6,000 individuals. And the reason I use this is because you can more clearly see the pattern here. So first we look at the causal effect test, whether you can identify the genes that are potentially causally associated with the trait. And on this plot on the left, and in WTCC, we have 7 different diseases. And I'm picking up 2 example ones. One is bipolar disease, the other is type 1 diabetes. The reason I pick up these 2 is because bipolar disease is very highly polygenic trait. So with 5,000 sample size, you don't really expect to see anything. So therefore, the bipolar disease serves as your null. On the other hand, T1D type 1 diabetes is autoimmune disease. It has very strong signals, even in the WTCC data. So that gives you sort of alternative trait. So you can treat the left as a null, the right as the alternative. And we compare a bunch of different methods in terms of testing the causal effects. So this includes our method, is the red-colored one, and a bunch of other different methods. And you can see that on the left, the red-colored one sits on the diagonal, means that you do not detect any signal in bipolar disease, which fits your expectation. It's a hard trait. With 5,000, you wouldn't expect to see anything. So that's why it sits on the diagonal. And each dot represents 1 gene. So we analyze about 20,000 genes. You have 20,000 different dots over here. While on the other hand, you can see the other different methods. Some shows inflation, some shows deflation. So this one is SMR, shows deflation, meaning that it doesn't really have much power. On the other hand, the other one shows inflation, and that's kind of expected because if you don't control for the horizontal pleiotropy effects, then that horizontal pleiotropy effects is going to be attributed to a true signal. So therefore, you tend to see 4 signals if you don't control for that. And you can clearly see those 4 signals by the ordinals, especially this, the black-colored ones for the bipolar disease. On the other hand, for type 1 diabetes, that represents an alternative. And in the alternative, you can see that our method at the beginning sits on the diagonal very well, but then you see a huge deviation indicating that you actually have power to detect signals for type 1 diabetes. So that's a GWAS and WTCC study. So that's on causal effect testing. And remember that the new method we have, the PMR, also allows you to test for the horizontal pleiotropy effects. So which is very important. This allows you to really test for each gene, whether some genes have horizontal pleiotropy effects or some not. And for bipolar disease, you can see it sits on the diagonal. That's very nicely sits over there because you don't really have much strong signal here. While the other one shows you a huge inflation. And for type 1 diabetes, our methods, again, behaves very well, sits on the diagonal at the beginning, then you start to see signals, which is, again, consistent with the ability to detect horizontal pleiotropy effects in this type of data. And you can summarize these 2 different graphs into this summary across the 7 different traits. Specifically, we can show the genomic control factor. On the left, across the 7 different traits, you can see that our line sits around 1 and even slightly below 1, meaning that it's slightly conservative, but on the QQ-Pro looks really nice. And on the other hand, on the right-hand side, it shows the power, so the number of significant genes versus the 7 different traits. You can see that our method has also much higher power compared to other methods. So basically, it gives you a better control of type 1 error and higher power. And this is also similar pattern across the UK Biobank traits. So since our method applies to summary statistics, so it's easily scalable to UK Biobank. So you can see that our genomic control factor is pretty decent here, and our power is also pretty high compared with a range of different methods. So that's our PMR framework. So the key message here, the PMR, is that you control for horizontal pleiotropy effects, so that you can calibrate, get better type 1 error control, while you also accounting for the uncertainty so you can get better power. So the PM framework is very nice. So we built upon the PMR framework. We did a bunch of different extensions. So the first extension we did was to extend the TWAS analysis towards modeling multiple outcome traits, and those multiple outcome traits are potentially correlated with each other. So specifically, this graph is very similar to our previous graph, except that we have a gene expression, except that one trait now you have k different correlate traits. That's because in biological settings, we don't really just analyze one trait. We actually collect a bunch of correlate traits. You can imagine that, say, if you want interested in blood lipids, right, you have HDL, LDL, total cholesterol, triglyceride, and also if you want to measure height, sometimes you also measure weight, and you want to model them and join them together because there's certain SNPs or certain genes may have this pleiotropy effects that are associated or affects multiple different traits. So therefore, it helps to model all those biologically correlated traits together in this joint multi-outcome probabilistic Mendelian randomization framework. We call it moPMR. So that's our framework. We try to model multiple outcome traits. And modeling multiple outcome traits is very beneficial. And that benefits really depends on how the traits, they are correlated with each other and how the correlation is related to the gene effects on those individual traits. So we decide to look more carefully to figure out different settings to see in which setting you can actually gain power and in which setting maybe you won't expect to gain power. So we focus on two traits because it's easier to see. So we look at Y1 and Y2, there's two different traits. And we divide them into 6 different settings. So for example, in the first setting, so Y1 in the top case, Y1 and Y2, they are positively correlated with each other. And then the gene expression have a positive or consistent effects basically onto Y1 and Y2. Or alternatively, maybe Y1 and Y2, they're negatively correlated with each other. Then also the gene has the opposite direction on Y1 and Y2. So in that case, in either of those cases, you can see the power of performing multivariate analysis is much, much higher compared with univariate analysis. Basically, gain a power like 2 to 3 times for the increase. And those are rarely see in GWAS studies because typically if you develop methods, you only see 5% power gain, 10% power gain. So therefore, by modeling multiple traits, you can actually gain really substantial power gain over here. And in other cases, even though Y1 and Y2, for example, these two are not correlated with each other. But as long as the gene expressions have effects on both of them, then by modeling them jointly, you can actually borrow information across this to gain power as well. So the power gain is not as substantial as the first one, but it still gain quite high power, so 50% of power gain. But certainly in certain cases, especially say the last case, Y1 and Y2, they're not correlated with each other. And X is not affected with both, or Y1 and Y2, they're correlated, but X does not affect both. This represents a null setting. In the null setting, you can see that our power is controlled, well controlled at this dashed line. So indicating that our methods still works pretty well in controlling the type 1 error. And then in certain settings, especially the scenario 3 and the 5th scenario, I think our actually lose the power a little bit. So those are the cases where these two traits are not correlated with each other, but gene expression only affects one of them. And so this is really a univariate case. And in this case, you want to run univariate analysis. So you can see that we lose about a small percentage of power, maybe 5% of power, but it's a small percentage. So it still was running a multivariate analysis since in any real data set, you don't really know what's the underlying truth. So, yeah.
So here, when you do the univariate analysis, you have 2 p-values.
2 p-values,
So
yeah.
you do the Bonferroni correction.
We do the minimum p-value. I think we just pick up the minimum p-value and do Bonferroni correction. Yeah. Yeah.
Okay.
Yeah. Mm-hmm. Okay, so you can see that that's the benefits of modeling multiple traits together. So that's our first extension. And that's a moPMR. The second extension we did was motivated by a TWAS collaboration study we did with our local collaborator, Jennifer Smith. So she has this Genova data cohort. And this cohort, she has collected gene expression data for 1,000 African Americans and for 800 European Americans. So we have two different ancestry in that particular data. And we measure the SNPs, we measure their gene expression, we identify SNPs that influence gene expression to identify those eQTL. And so for those eQTLs, we try to see what's their effect size similarity in African Americans versus their effect size in European Americans. So each dot represents one eQTL, and the x-axis shows their effect size in African Americans, y-axis shows the effect size in the European Americans. And you can see that they tend to see a consistency between these two populations, which is kind of expected because you might expect the underlying biology, the cis eQTL regulation and gene expression are going to be consistent across different genetic ancestries. So therefore, motivated by this, we decide to extend TWAS towards analyze multiple genetic ancestries. Specifically, we developed a method called METRO. Now it's extending built upon the PMR framework, but now this allows you to incorporate multiple genetic ancestry of eQTL studies into the model. So the intuition is that if your GWAS is a European ancestry, right, you are incorporating African ancestry eQTL mapping studies, that's going to improve your sample size. So therefore, that's going to improve power. And such power gain is going to be especially obvious in African American GWAS studies, because in African American GWAS studies, we already tend to have a small data set. So therefore, any gain by incorporating the sample size of eQTL is going to be substantial. So in fact, that's what we see here. So I'm going to show you 3 different data sets. So one is BMR sample from the GIANT study, BMR from AAGC, and also type 2 diabetes from media study. Those 3 are all African American GWAS studies. And if you simply run any other methods using just our European sampled eQTL study, you will see that the power are okay in the first study because it's a GIANT, you have a large sample size, you tend to see signals. Each dot is again a gene. And but in the middle one, it's very hard to detect anything. In the last one, none of the other methods detect anything. However, if you are able to use METRO, which incorporate multiple genetic ancestry into your model, this time you can see the black line is our method. You get much higher power compared with all the other methods. And this is very obvious over here. We detect 200 genes using our METRO method by incorporating multiple genetic ancestries, and it's much higher compared with others. And our method is the only one that's detected any signals compared with all the other methods in the last study, the media study, which is a relatively smaller type 2 diabetes study. And the top gene we identified, one of the top genes is MAPT, which is a microtubule-associated protein tau. It turns out that gene is kind of well known for association with type 2 diabetes. It belongs to the family of microtubule-associated proteins. It stabilizes the microtubule tracks. It's hypothesized to play a critical role in the glucose-stimulated insulin secretion. So you can see that the top signals all actually make quite good sense from this multi-ancestry study. So that's our second extension. We extended towards analyzing multiple genetic ancestries. The third extension we did was to incorporate multiple gene expression prediction models. So that's a setting where, if you recall early on, PredictScan uses the elastic net to do a SNP on gene expression prediction. Then the TWAS uses the BSLMM to predict SNP effects on the gene expression. And naturally, you are wondering, given there are so many different prediction models, we also have a lasso, we also have other prediction models. Which model should we use, or should we use all of them? So it turns out that you can actually apply all of them, and you can aggregate all their p-values directly. Specifically, what we propose here is the method called HMAT, which corresponds to a harmonic mean p-value aggregate TWAS. The approach it does is that it's for each gene, at the time you apply for multiple gene expression prediction models. And for each gene expression prediction model, you'll get p-values. So if you apply for k different gene expression prediction models, you're going to get k different p-value for this particular gene. And then we aggregate all the k p-values by computing their harmonic mean as their outcome, the final p-value for your analysis, for your TWAS. So that works pretty well. It actually accounts for their correlation structure among the p-values, and it's very stable, and the results are very well behaved and gives you more power compared with using any single expression prediction model. The intuition is that for one gene, maybe the prediction model A will work better. For the other gene, the prediction model B will work better. By aggregating all the prediction models, then your aggregation approach here will be better for every single gene. So that's our third extension over here. And then the last extension we did was to extend it towards TWAS fine mapping. So that's our so the main idea is this. So so far we have been introducing this TWAS analysis framework as always marginal in the sense that we analyze one gene at a time. However, in a local region, you always have multiple genes, and those multiple genes they tend to share similar gene expression correlations. Their expression correlate because they share sort of cis regulatory components. They are in a similar region. They probably have a similar open chromatin and this level in that local region. So therefore, the gene expression are correlated with each other. And second, they share similar cis SNPs. So therefore, their TWAS association evidence are going to be similar to each other. So in fact, if you simply just run a standard TWAS analysis across, say, the 6 different genes, you will see that if the truth is this red one, some of the nearby genes will also show relatively strong p-values compared with the others. And this is very similar analogous to the GWAS association analysis. In the GWAS, if you just do a marginal GWAS analysis, you're going to see a lot of signals in the middle part that's nearby the truly causal SNPs. So therefore, you will see that you tend to see a lot of local TWAS signals. And the goal is instead of picking up all the top ones, rather you want to ask which one is more likely to be causal, which one is actually causal. So therefore, we decide to extend the PMR towards the TWAS, the TWAS fine mapping analysis. And we developed a method called GIFT, which is basically the gene-based integrated fine mapping through conditional TWAS. It's doing a conditional TWAS analysis. So the main idea is this. So in the TWAS setting, in the local region, we actually compare with the GWAS fine mapping. The GWAS fine mapping, it turns out to be a much easier problem because we actually only have a few different genes in the local region. Typically, the number is about 10, 20. Occasionally, for very large ones, you have 100 or 200. But if your number is only 10 or 20, this identifying the truly causal one is actually turns out to be simple because all you need to do is to do a conditional analysis. You can test this gene conditional on everything else, or the other genes, by incorporating the other genes as a covariance in your model, and get your conditional p-value. And do the same thing for the others, do the same thing for the others. And the intuition is that if this one is not a causal, then by conditional on everything else, you're basically conditional on the causal gene, then you wouldn't see signal for this gene. On the other hand, if this is a causal gene, then controlling on everything else, you are still going to see signal. So therefore, through this conditional analysis, you would be able to really identify the truly causal one in the local regions. This allows you to do to perform the TWAS fine mapping. And so this is our GIFT method, the main idea. And this method can use both individual level data or summary statistics. And it's really doing this really conditional analysis while we still account for the gene expression prediction uncertainty. So the hope is that now, if you perform just previous marginal analysis, you're going to identify, say, 3 candidate genes in the local region. While through conditional one analysis using GIFT, now you only identify the one, allows you to narrow down what's the truly causal gene associated with that trait. And this GIFT is extremely effective. So we applied it to UK Biobank. So you can see that our method, first, if you look at the QQ plot, our method is quite well behaved, especially compared with other methods. So you can see that it sits on the diagonal at beginning, indicating it's calibrated type 1 error control, and then it shows power identifying. And also, in addition to that, the ability of GIFT lies in its ability to really narrow down what's the causal gene in the local region. So the way we can check this is this. So for each region, right, we can ask how many signals you have by our method. So for the majority of genes, regions, you can see that over 90% of regions, you only identify 1 signal. While for some regions, you identify 2 different signals, meaning that for the majority of regions, you are able to narrow down to a single gene as a potentially causal gene. Or for other methods like focus, about half you are able to narrow down 1, but for the other half, you can't really narrow down to 1. You can only narrow down to 2 or maybe 3. And then the other methods are even worse. Sometimes you identify 4 or 10 different genes. Those are basically, you don't really have the precision to identify the truly causal ones. So therefore, you can see that in the UK biobanks, it's extremely effective in fine mapping the sort of the truly causal genes here.
Here you analyze 6 traits.
We analyze 6 traits over here. Yeah, I think that's a summarization of all the 6 traits. So that's our sort of PMR framework and a multiple extension towards PMR. And PMR is really based on the Mendelian randomization analysis, the causal inference framework. So when we were developing this sort of PMR framework, there was one day and we tried to write down equations and we realized that there's actually an alternative view of how TWAS is working. And this alternative view really changed our entire perception of how TWAS is actually doing. So here's what we see. So let's take another look at TWAS. So as I mentioned earlier, TWAS is 2-step procedure. It can be described in this following 2 different steps. In the first step, we look at equatorial sample. So you build a SNP predicting the gene expression. Then in the GWAS sample, we basically plug in your beta-1 hat, the SNP effects on gene expression plug in there. And you test the association between Y and this plug-in term. This is the GWAS term. So you want to test alpha. So that's the TWAS or predicate scale in this 2-stage framework. Now it helps to rewrite it slightly differently. So it's the same equation except that instead of beta-1 hat, I simply call it beta-2. So the beta-2 represents the effects, the SNP effects on gene expression. And the beta-2 over here is really modeled in the TWAS. It effectively assumes beta-2 is the scalar alpha times beta-1. So beta-1 is p vector, beta-2 is p vector representing p different SNP effects on the gene expression or the trait. And the TWAS assumption is basically to say that beta-2 is a scalar multiplication of beta-1. So it just scales the beta-1 to get your beta-2, and that particular scale is your causal effect size. So once you realize this, then you realize that the TWAS makes this huge assumption. It says that the SNP effects on the trait is scalar multiplication of SNP effects on gene expression, meaning that the latter is fully informative testing of the former. And this assumption appears to be very strong because in real data analysis, people found that only 11% of genetic effects are really mediated through gene expression, not 100%. While TWAS is making the assumption that 100% of SNP effects are mediated through gene expression. So that concerns us. So we decide to consider whether we can really generalize this. And one step forward, it turns out that if you are able to make additional model assumption on beta-1 through, say, linear mixed model assumption or Bayesian sparse linear model assumption, then for each SNP-J with non-zero effects, the TWAS assumption you can also retain in this way. So you have the J-SNP effects on gene expression, J-SNP effect on the trait. And the TWAS assumption basically says that it follows a bivariate normal distribution, and their correlation is 1, either 1 or -1. So the effect size, they are correlated. So the effect size on gene expression and the effect size on the trait, they're correlated, they're perfectly correlated. The correlation is either 1 or negative. That's, again, this huge assumption there, but that's what the TWAS makes the assumption on. And TWAS basically aims to test whether alpha equal to 0 or not. And in this case, it's also equivalent to testing that whether that variance component sigma beta square is 0 or not. Now, once we arrive this stage, this equation looks very familiar because that goes to a scat assumption. Scat is never used for integrating EQTL mapping studies with GWAS. Scat is primarily used for rare variance tests. And scat in this particular setting is effectively to say that this two also follows a bivariate normal distribution, but their correlation is exactly 0, meaning that the SNP effects on gene expression is not correlated with the SNP effects on the trait. So that's why in scat model assumption, all you care if you want to test the sigma beta squared, you can throw away the gene expression, you only focus on GWAS and to do this particular test over here. So, so this, I think it helps to take a quick recap to see what we really observe through these equations. So it looks like both GWAS and scat, they are basically doing something similar. They want to test whether sigma 2 square equal to 0 or not. So that's the SNP effects sigma beta 2 squared. That's the SNP effects on the trait. And in doing so, they are making very different assumptions on this correlation parameter R. Specifically, TWAS sets are equal to either +1 or -1. Basically says that EQTL study is fully informative for the testing in the GWAS. So therefore, we need to throw in our EQTL mapping study. We may need to make sure that it's scalar multiplication of the beta 1. On the other hand, scatter sets are equal to 0. Basically says that EQTL is not informative. So therefore, we throw away the EQTL mapping study. We focus on our GWAS alone and we do the analysis. So naturally, it depends on whether the assumption holds for any particular gene, right? So their power depends on whether the assumption matches the truth. And this is unknown. And this also will vary for every gene and every trait, because for some traits, maybe R is close to 80%, 90%. For some traits, maybe R equal to 10% or 20%. So it really depends on varies across genes and varies across traits. So therefore, we decide to take a step back and create a more general approach that includes both the SCAT method and also the TWAS method as spatial cases. So this is a method we call vintage, or variant set test integrated TWAS for gene-based analysis. So it builds upon the affirmation framework. So you still have two different equations. You still have beta 1, beta 2, and still bivariate assumption. But instead of setting this either to be +1 or -1 or 0, now we try to infer this. We have R over here. We have our correlation parameter inside. And we're going to estimate our correlation parameter for every gene expressed.
So you know that R is either 1 or -1. Why do you estimate?
Because we don't know what R is. When R is 1, basically means that the SNP effects to the trait is 100% going through gene expression, which is a very strong assumption. On average, I think it's only 10%. So the average of R should be like 0.1 or something like that. Yeah, that's a great question. So therefore, we generalize this. We try to estimate R, and we also want to test R. And then the R will also allow us to borrow information, properly borrow information from your EQTL study into the genetic test. So by modeling R, now vintage effectively includes both SCAT and certain versions of TWAS as spatial cases. You can imagine by R equal to 0, it goes to SCAT. By setting R equal to +1 or -1, it goes to TWAS. And R is an important parameter that captures the amount of information available in EQTL study for genetic association tests and TWAS. So therefore, inferring R is important. It allows you to ensure the robustness of the test. And also importantly, biologically importantly, R turns out to be closely related to mediation analysis. It quantifies the proportion of SNP effects on the trait that's mediated through gene expression. So therefore, inferring R turns out also to be biologically meaningful. So that's the vintage modeling framework. And what vintage does is to do two different tests, a genetic association test, just like TWAS and SCAT, it's testing sigma 2 squared equal to 0. But also include this genetic correlation test, testing whether R equal to 0 or not. And both tests can use either individual level data or summary statistics. And I think the former, getting a calibrated test for the former turns out to be quite tricky. We spent almost 2 years on that, figuring out how to do the proper test. And eventually we'll get a decent solution for that. So that's our framework. So it includes both TWAS and SCAT as spatial cases. It's able to use summary statistics, and it's able to interpret. Now I show you some sort of intuitive results here. So here's the simulation. What I'm showing here is the power versus the genetic correlation parameter. So if you set genetic correlation parameter equal to 0, recall that our method reduces to SCAT. And so in this case, you would expect that SCAT actually has a high power. So SCAT is this one. The SCAT is a green line. So you can see that the SCAT power is pretty high at 0, and it's actually consistent across different correlation parameters, basically a line over here. That's because SCAT assumes the genetic correlation is 0. It throws away your EQTL mapping study. So no matter what the choose, it's always just use GWAS. So therefore, it's stabilized. It's a horizontal line. On the other hand, TWAS makes the assumption that it's either -1 or +1. So therefore, if the choose is indeed -1 or +1, then you have the highest power. However, if the choose is not, then you can see the TWAS power sort of reduces and into the middle. So the lowest in the middle. While our method, vintage, is really the best of the both worlds. You can see that it's more powerful than either of those approaches across a wide range of potential genetic architectures. That fits your assumption. And so that's very important. And the second important thing is that you will see that for TWAS, you actually tend to see a lot of false signals. Specifically, in the absence of mediation, if the chooses I is zero, if the chooses that the SNP effects on gene expression is not correlated on the SNP effect on trait at all, the SNP effects on trait is not mediated through gene expression. In that case, you can see that TWAS actually still have power. So this is a TWAS line. Even though if in choose is zero, TWAS is going to give you signal. And this is expected because TWAS from our framework, you can see that it's really just doing a genetic variance test. However, this is also unexpected because in the entire field, everyone is interpreting the TWAS signal as potential mediation and results. So, therefore, this dovetails with the discussion on the TWAS inflation issues. TWAS tend to give you a lot of false signal in the real data sets. And in addition to those so far focus on genetic variance tests, but we also have this genetic correlation test, which really allows you to identify genes that are potentially mediating the effects. You can see that our method is also has calibrated type 1 error control, which is underneath this red line in the middle when the choose is zero, and while the choose is not zero, then our method has power compared with other methods. And it works well on the sparse structure. And so I'll just go through the real data application. So we applied to EQTL gene EQTL study, which contains, I think, 30,000 individuals, about 11 million SNPs. And we also look at the UK Biobank, which contains 13,000 or 14,000 protein coding regions. And we analyze 18 disease related complex traits. And then we use the European once on the genome project as the elder. So here are basically showing the results. So on the left, it shows the number of identified genes by vintage versus heritability. You can see that with increasing heritability, we tend to identify more genes, which is consistent with your expectation. One thing that surprises us was that, on the right-hand side, so we're showing the power versus 3 different methods, the power of 3 different methods across 18 different UKB traits. One thing you can see that our is more powerful than the other 2, which is expected. However, what's unexpected is that scattered actually works pretty well, indicating that scattered is actually perhaps better method than TWAS at this. So maybe TWAS should not really be used in the field since the scattered actually works better. And the reason scattered works better is because for most genes, the gene expression mediation percentage is very low. So you can see that across 18 traits, the estimated R is always concentrated around 0, meaning that the estimated R, I think the average was around 10 to 12%-ish, very consistent with the previous literature, indicating that the scatter is perhaps a better assumption, makes a better assumption than the TWAS does. And so vintage is, and for the genetic correlation test, vintage is only effective method identifying 61 signaling genes, while other methods only identify 0. And vintage estimate mediate proportion of mediation effects gene inflection is about 12% across all traits. Again, very consistent with the early study on similar traits. And I'll just show you 2 examples. One is the KMK1D for SBP. So in this case, the genetic covariance test is significant, genetic correlation is significant, and the proportion of mediated through this particular gene expression is 65, meaning that the SNP effects on the SBP, 65% of SNP effects is mediated through this particular gene. And on the other hand, another example shows you that this is significant in the genetic test, but not in the correlation test, meaning that the SNP effect on the trait is potentially at least not significantly mediated through gene expression. So that's very helpful. That gives you really useful biological information on teasing up the actual the transcriptomic mechanism underlying those associations. I'll skip that on. So that's, I think, all I want to talk today. And as a summary, you can see that we talk about PMR, which views TWAS from a Mendelian randomization causal testing framework, but it controls for gene expression prediction uncertainty and test control for horizontal pleiotropy. And we extended them to analyze multiple genetic ancestry, multiple outcome traits, multiple gene expression prediction models, and finally also fine mapping as well, TWAS fine mapping. And we finally also have the vintage framework that unifies the TWAS and scatter into the same conceptual framework and give you additional information. And all those work is really done by my talented students and postdocs, and I think 3 of them are now faculties elsewhere, and one is hopefully wrapping up and will leave either this year or next year. And we are all the research are supported by NIH grants. And thank you so much for your time, and happy to answer any questions you have.